Good evening and welcome to tonight's event. My name is Gareth Gaskell. I'm a professor of psychology at the University of York. And it's my pleasure to introduce tonight's speaker. So now to our speaker tonight, it's a great pleasure to introduce William A. Harris. He's Professor Emeritus of Anatomy at the University of Cambridge, UK. He's a Fellow of the Royal Society and co-author of several books, including The Development of the Nervous System and Genetic Neurobiology. He's also co-editor of Retinal Development. So without further ado, over to you, William.
Well, thank you very much. I hope you can hear me. Thank you for being at the York Festival Ideas, here to listen to me talk about how the human brain is built, which is the subtitle of my book, Zero to Birth. The brain, as you all know, is the organ inside our skulls that perceives, thinks, imagines, that gives us humanity, personality, insight, wisdom, etc., etc. And most books about the brain focus on some aspect of how it works, which is excellent. And it's true that we still don't know a whole lot about how it works. But this book, the one I wrote, focuses not on that, but on how it's built, on what the instruction manual for building a brain might look like. And again, there's much we don't know, but this book tries to cover the basics of what we do know. And it's published by Princeton University Press, whose mission is written here. And I thought this is kind of interesting. Princeton University Press brings scholarly ideas to the world, publishing peer-reviewed books that connect authors and readers across spheres of knowledge to advance and enrich the global conversation. Now, my thought was that most people don't know much about how a brain is built, and that's reasonable because most people haven't had courses on the topic at university. But it's interesting, and people want to know things like, how is the most complicated thing in the known universe built? And what is going on in the womb in a baby's brain? And what medical conditions are linked to what steps in brain development? And I think this topic fits well with the York Festival's mission, which is to educate, entertain, and inspire, which is what I'll try to do in the next little while. Now, my research on this topic started at Caltech in 1972 as a graduate student working on flies. But over the years, over my career, I have worked in different universities and worked on salamander embryos, frog embryos, fish embryos, mice embryos, etc. And I also taught courses on the subject for 40 years at University of California, San Diego, and in Cambridge. And with co-authors, as Gareth mentioned, Dan Saines and Tom Ray, we wrote a textbook, The Development of the Nervous System, which is basically for senior undergraduates and graduate students, now in its 4th edition. And when I retired, I decided to write Zero to Birth, which is based on the same basic info as the textbook. It's the hardcore basics, but it's aimed at a broader audience. So it's more approachable. And one of the ways I tried to make it more approachable is to talk about it like the cells in your brain have their own lives. And it's a biography, if you want, of the component cells of your brain. And it's full of cellular drama, some life and death struggles, as you'll hear a bit later. And it also includes more about how the discoveries were made and the stories of the scientists behind the discoveries. But let's get stuck in. I'm going to give you a couple bits of the book. So here's a mom playing with her baby girl, staring into her eyes and saying to the baby how amazing she is. And I'll quote you from the introduction. The newborn baby not only possesses her own fingers and eyes, she also has a mind of her own inside her skull where the mother cannot see it and marvel yet further. And although we can't see each other's brains, and even if you could, what would it look like so much? Wouldn't tell you too much. But a newborn baby's brain is an anatomical wonder of internal complexity. And it's already super active, taking in information, learning about the world that it's been born into, controlling its wiggling fingers, and so forth. And if you do an MRI of a newborn baby's brain, it looks already very well formed. If I were to show this to some of the students I taught, medical students I taught neuroanatomy to, I doubt if many of them could be easily tell the difference between a newborn baby's brain and an adult brain. Now, I'll give you a little background that you'll need for the rest of the talk. We probably already know that your brain is mainly composed of two cell types, neurons, and this here, if you can see my pointer, this big yellow cell here is the neuron, and glia. These blue and red cells are the glia. The glial cells have roles in the development and function maintenance of the brain, but the mental activity of your brain, your thoughts, your sensory experiences, your dreams, and your coordinated movements are due to the neurons. And a typical neuron in your brain receives electrical inputs on some of its branches. So here's a typical neuron, and these are the branches that it receives information in on. It gets input onto these branches, which are called dendrites. And it's a bit like a computer chip. It does computations based on these inputs, and it sends out the results as electrical impulses that travel along another extension of the axon, of the cell, of the neuron, called the axon. And this is the long axon, a long kind of cable, electrically active cable. Notice that there's many dendrites, but only one axon. But at the end of the axon, it branches, it makes an arbor. And this is where this cell puts its output onto many other neurons, and possibly in a different, long, distant area of your brain, and those neurons do their own computations and send out their own signals, etc. So the connections between neurons from one axon to the dendrite of another are called synapses, and you've probably heard that word. And this is an electron micrograph of a couple of synapses. This is the terminal of an axon, and it's filled with vesicles that have neurotransmitter in them. And this is the dendrite of the receiving neuron. So the signal is going from this cell to this cell, or this part of a cell to this part of a cell. And I'll just show you a human neuron here. And this is a recent reconstruction, amazing, from Jeff Lichtman and his colleagues at Harvard University. And you can see in this cell here, the dendrites. This one little process is the axon. These are the dendrites. And all these little red and blue things here are the synapses on this cell. And just look at it. I mean, there are hundreds. Here's the piece that he reconstructed, just 1 cubic millimeter of human brain tissue. And there's 150 million synapses here. So now we can talk about one of the central issues in brain development, which is how the brain gets wired up with such precision. And this is covered mostly in chapter 5 of the book. So we're skipping chapters 1 through 4. Now imagine a human brain during the latter half of maybe the first trimester of development. Developing neurons are sending out these axons that are traveling simultaneously through the brain, heading to destinations far and wide. The histologists of the 19th century knew that neurons had long axons, but it wasn't possible at the time to trace them or to look at the developing brain with such techniques as this one, which is an MRI technique, and watch the neurons grow. But then 134 years ago, in 1980, the great Santiago Ramon y Cajal, who is considered to be the father of modern neuroscience, made a significant breakthrough. And here he is 10 years earlier, near the beginning of his career, and this is his lab. And trying to, there we go. He looked down the microscope at the spinal cord of a chick embryo, and he saw enlarged endings at the tips of what he presumed were growing axons. So here are some cells up here. This is the cell body, and they're sending axons, and you can see it at the tips of these axons. There are these little enlarged areas. And in his memoirs, he wrote, "I had the good fortune to behold for the first time that fantastic ending of the growing axon. In my sections of the spinal cord of the 3-day chick embryo, this ending appeared as a concentration of protoplasm of canonical form, endowed with amoeboid movements. In this curious terminal club, I christened the growth cone." So that's the growing part of the axon. And it's amazing that he could think of it as amoeboid movements, because he only had still slides that were of fixed dead material. Now, we know that growth cones power the extension of the axon and help it navigate to its distant destination accurately so that the brain gets wired up properly. And a huge breakthrough in how this happens, this correct wiring up, was made in the 1990s by this fellow, Corey Goodman, when he was at Stanford University. And Corey looked at the problem of how axons navigate in the embryos of locusts, because locusts have large identifiable neurons when they're embryos. And Corey and his colleagues were able to look at particular neurons as they made navigational decisions. And here's an example of his work. Here's the growing tip of a neuron called G, and it's crossing the midline by associating with another axon from a cell named C. And they bundle together. But once G crosses the midline, it lets go of C and grabs onto another axon of a previous axon and gloms onto it and heads up north towards the brain. Whereas C grabs onto another axon and heads south down towards the tail of the animal. So when axons stick to each other like this, they form what are called fascicles. And this selective sticking is part of the answer to how the brain wires up properly. And through very clever genetic and molecular work described in Chapter 5, Corey Goodman and his colleagues found a set of selective adhesion molecules. And he called them, because axons make fascicles, he called these molecules fasciclins. And they turn out to be molecules that selectively stick to themselves. So this cell, this is a fasciclin molecule that's stuck in the membrane of one cell, and this is the membrane of another cell. And they're both expressing this red fasciclin molecule, sticks to each other. So cells that are expressing red fasciclins stick to other cells that are expressing red fasciclins. Green sticks to green, blue sticks to blue, and so on. And the idea that emerged from this work was that most axons travel to their destinations in a way that's a bit like how Bostonians travel to their destinations. If they're going to the zoo, they take the green line to Park Street, get off there, switch on to the red line, get off at Ashmont. From Ashmont, they take the the 22 bus to Franklin Park, where the zoo is. And as more and more axons are added, they They go along these labeled lines, and when they get off the final bit, they may add a new fascicle in for others to follow. So the simple scaffold of the first axons becomes an increasingly complex set of major and minor pathways through the brain. But now I want to talk about those very first axons, the pioneers who navigate in the developing brain. And these are some of them. And this is a drawing by Santiago Ramon y Cajal of some of the neurons in our eye that send out pioneer axons to the visual areas of the brain. And this was an issue that fascinated me and my wife and scientific collaborator, Christine Holt, whom I'll talk a little bit more about later. Pioneer axons can't follow others because they're the first to form the pathways. So how do they navigate is what we wondered. And here is a microscopic reconstruction from the brain of a frog embryo of a single retinal cell. And this is the retinal cell here stained in black. And you can see its axon going in and up here like this. This is the retina here. So, and these are the dendrites of this cell. So it's taking in visual information from the retina, and it's sending out an axon, goes out of the eye, along this structure here, which is the optic nerve. It enters the brain here in the ventral, the bottom of the brain here, which is called the optic chiasm. And then it grows upward in the brain, dorsally, as we like to say, towards its targets. It's a long route. And the question is, how does it know how to go take this route? And here's, this will be interesting. This is a movie that Christine and I made in the 1980s of the first movie of axons growing in the brain. And hopefully you can see these retinal axons growing in the brain with their growth cones at the tips. And they're growing they grow in an error-free way. And when they get to their targets, they start branching like this. So what is the
brain growing until mid to late childhood?
Sorry.
Stabilizes in size for
one possibility was that the target, the midbrain, secretes a diffusible come hither molecules. And then the growth cones, like bloodhounds, follow the scent to its source. This idea of long-range guidance was very popular in the 1990s. And Jeremy Taylor at Oxford University did a very revealing experiment. What he did was he removed the target area from a developing frog embryo before the pioneering retinal axons had grown out of the eye. And guess what? The retinal ganglion, the retinal axons still navigated beautifully all the way there, only to find that their targets had been long gone. Damn. So the long range attraction molecules for pioneers began to seem very unlikely idea. The other possibility was that, local cues were involved. And to test this idea, we took out a little piece of the embryonic brain tissue that was en route for retinal axons and rotated it either clockwise 90 degrees or counterclockwise 90 degrees and put it back, and then watched what happened when the retinal axons grew into this rotated piece of tissue. And what happened is shown on the slide, basically, was that when these axons entered the piece of the rotated tissue on their way, they also turned, as though they were paying attention to the local cues in the rotated piece. But when they exited the rotated piece and discovered where they were, i.e. they were not in the place where they expected to be, damn, they had to make corrective turns to get to their target. And these experiments suggested that pioneer axons navigate by reading local cues rather than sniffing out distant targets. Now, in the early 2000s, many labs around the world started to discover what these local cues were, and they found attractive ones and repulsive ones. These are called guidance cues for growing axons or growth cones. And here in this top two panels here, there's a little pipette that's putting out an attractive cue, and you could see the growth cone turning toward the pipette. And the bottom two, here's a growth cone that's being exposed to a repulsive guidance cue, and you can see it turning away. Now, one attractive cue for young retinal axons is called netrin, and it's one of the very first discovered guidance factors, and it's Sanskrit for the one who guides. And it's right in the center of the retina. And axons, as you see from the retina that are going out towards the optic nerve, come to the center of the retina. Netrin helps them guide their way out of the eye. Here's another guidance cue, and this one is at a place I mentioned before, the optic chiasm. And this molecule called Ephrin B, you don't have to remember any of these names or anything like this, just they have names. And in the book, I try to minimize the names, just give you a few examples, but you don't have to memorize them. The concepts are what's important. This cue here is attractive to the green axons, which grow towards then across, but it's repulsive to the red axons, so when they get close to it, turn away from it. It sits at the optic chiasm, where some axons from your eye cross and some axons don't cross to the other side of the brain. The importance of the optic chiasm was understood by Isaac Newton in the early 1700s. And it basically is to unite the visual world from both eyes. So here's a human brain, you can see. And in this left eye, the right side of the left eye in blue here crosses over to the other side of the brain, whereas the right side of the right eye doesn't cross over. It doesn't cross over the chiasm, and so it stays on the same side of the brain as the right side of the left eye. So the left visual world that falls on the right side of both windows ends up in the left, in the right visual cortex, and the right visual world ends up in the left visual cortex. And that's all down to this selective crossing at the chiasm. So it turned out that even though the early brain looks morphologically rather featureless, it is actually highly patterned molecularly with guidance cues for axons. And here are just some of those that are relevant to the retinal axons that are growing as pioneers in the brain. One of the other things I started doing as a retiree was painting. And here is a painting of a growth cone that I made, and I'll read from the book. Growth cones are sophisticated readers of molecular maps in the developing brain, continuously integrating combinations of attractants, repellents, cell adhesion molecules, and other potential guidance cues when making navigational decisions. And like axons, sometimes people go to place A and then they go on from place A. They're attracted to a place and then repelled. They don't want to stay there. And when I explain this to students, I often talk about the Worthington services stations on the A1, which look very attractive when you're hungry and you're nearly out of gas. But once you get there and eat the food and so forth, you really want to get going again. And it's the same with growing axons. And here's another reading from the book, slightly longer. The mid-19th century saw wagon trains of weary travelers moving west across America, over the Rocky Mountains to Oregon and California. And most of these pioneers of the West first headed towards a huge granite hill shown here called Independence Rock in the Sweetwater Valley of Wyoming. Independence Rock marked the approximate midway point for those on the Oregon Trail. And here the pioneers often stopped for a while to rest and get prepared for the next stage of their journey. They often scratched their names into the rock, along with the dates they arrived, but they could not tarry long, especially if they arrived there at the beginning of July, where they might not make it over the Rocky Mountains before the snows came and made the mountain trails impassable. Well, growth cones have similar issues. They may also be attracted to a place en route, but they must not stop there for long. This is merely an intermediate target. Axons must leave such attractive intermediate targets and move on to take the next leg of their journeys. And we know that whether they are pioneers, trailblazing new routes, or using other axons as a kind of public transport system, growing axons frequently change and update their priorities as to where to head next. And how they do these updates is one of the newest and most exciting areas of research in this area. But other than for its own fascinating sake of discovering how the brain wires up, or part of how the brain wires up, this topic has medical relevance. Take, for example, spinal cord injury here, where the prognosis is very poor. Science has made serious progress in understanding some of the problems for repair of axons that are cut. Adult neurons seem to lose the mojo of their youth and have trouble making new growth cones and putting out new growing axons. And even if they do, the site of the injury is often full of repulsive guidance cues that they can't get through. And the molecular guidance system that was present in the embryo is no longer in place. And the distances are huge compared to when the pathways were first formed. And though this knowledge has, been very helpful, it has also shown how huge the challenge is. So that was all basically from part of chapter 5, wiring up. And each chapter has its own theme and relevant period of brain building. After wiring up, which comes firing up at chapter 6. And I'm not going to talk about chapter 6 much, except to say that each chapter, to orient the reader, and this was a suggestion of my editor, has a very short intro to the topic. So wiring up, which you just heard about, in which young neurons send out axons that navigate through the developing brain in search of specific destinations that are often far away. And firing up is in which we witness the momentous moment in the lives of two neurons when the axon of the first meets the dendrite of the second, and the two recognize that they are meant for each other. They stick tightly together and seal the deal with a synaptic kiss. So I was wondering, how to do this very short introduction that the editor suggested. And I used this in which way. And I thought it was really cool. And then was like about a year later, when I was reading Winnie the Pooh to one of my grandchildren, each chapter is Winnie the Pooh, in which Piglet does something or something like that. And I realized that's how I did that. Anyways, the making of synapses, which happens throughout most of the brain well before birth, is a huge turning point. Up until this point, building the brain has mostly been a task of construction, but once synapses are made, the balance shifts toward demolition. And the cell type that is most demolished are the neurons themselves. Adults like you and me have about 90 billion neurons in our brain, but newborn babies have more. They have 100 billion or more, and many billions have already died at the time of birth, and billions more will soon be lost in the first few years of a baby's life. Roughly half of all the neurons originally generated in the brain survive early childhood. And it turns out that much of the death of the neurons in the young nervous system comes from competition between neurons. And it's those neurons that make the most effective synapses that survive, and the ones that don't die. And perhaps this is a really good way to make a brain team with, neurons try out for positions and only the best survive. And that creates a good working and well-matched components in the brain. And in this context, I show here Nobel Prize winner Rita Levi-Montalcini over here on the left, and her mentor, Victor Hamburger, on the right. And while working on this problem of neural competition and survival at the University of Washington, they worked on this problem of neuronal competition and survival in the 1940s at the University of Washington. And it's really a fascinating story how before that, she was in hiding in Italy because she was a Jew hiding from German occupation and Italian persecution. And she was she was reading Victor Hamburger's paper and setting up labs in corners of her bedroom. And she made this discovery of cell death. And then later in Victor's lab, she found the first neuronal survival factors. And that's one of several survival factors that are essential for the survival of neurons. And it turns out that's what this competition is all about. Neurons need to get enough survival factors from their synaptic partners. And the less successful ones that don't get enough because they didn't make enough effective synapses die. So by the age of 5, you have about 90 billion neurons, just like an adult. In fact, the 90 billion neurons that you have are the same neurons that you had when you were 5 years old, and the same ones that survived from the time from your birth. And most of the neurons that survive the great period of competition and death are there for a lifetime, if you're lucky. And now I'll tell you one of the interesting pieces of information about how we know this. And it comes surprisingly from carbon dating the DNA in the brains, but it's been confirmed in other ways. So in the 1950s, countries began testing nuclear weapons above ground, and this released large amounts of C14, an isotope of carbon, into the atmosphere. And the levels of C14 rose accordingly in the DNA of all cells that were going through proliferation at the time, like the tree rings here. So this is 1000 BC or so. This is 1950. You see this sudden spike of C14 here. And if you look at the tree rings in 1950, there's not much going on here, but a tree ring in 1970, just after the spike here, has a lot of C14 in it. In 1963, the partial nuclear test ban treaty was signed. And since then, any nuclear testing has been done underground. And so the C14 peaked in 1963 and has been going down slowly since. So what you got really is a bomb pulse here. And this allowed Jonas Friesen, working in Sweden, to carbon date the neurons of post-mortem human brains who lived through the bomb pulse of C14. And what he found was that neurons of the cerebral cortex had C14 levels in their DNA that reflected the level in the atmosphere at the time of their birth. A person who was born near the peak of the bomb pulse and died many years later still had high levels of C14 in their cortical neurons that matched the atmospheric level at the date of the birth. While the cortical neurons of a person who was born before above ground testing began, even though they lived through all these times of high atmospheric C14, still had background levels of C14 in their cortical neurons, indicative of the low levels at the atmosphere at the time of their birth. And such studies indicate that cortical neurons that you're born with are not replaced throughout life, and that your neurons are basically as old as you are. And the really important thing here is that your brain is populated at birth with neurons that last a lifetime and are never replaced. And it's been known for centuries that brain injuries are severe and they're rather permanent. Now we're getting to the end here, and in the last couple of minutes, I'd just like to mention chapter 9, which is the last chapter of the book. And it's a bit different because it focuses attention not on a stage of the building process, but it explores the differences between human brains and those of other species, and how those differences arose. And I'll choose one story from that chapter, and this is the late Professor Miriam Diamond at UC Berkeley. She was particularly fascinated by the question of how postnatal experience affects the microscopic structure of the brain. And she was first attracted to this question by a story that a psychologist friend of hers told her about bringing home a pair of young rats as pets for his kids. And the kids adored the rats, and the kids liked the rats like the kids. The rats ran around the house. The kids played with them all the time, gave them challenges. And the psychologist, whose name was Donald Hebb, thought that these lucky pet rats probably had the benefit of an enriched and interesting life like rats in the wild, compared to their siblings that he kept back at the lab in cages, and one rat per cage. So he decided to challenge his children's rats against the lab-reared rats in a maze running competition. And the lab-reared rats turned out to be no match at all for the pet rats. And on hearing this story, Miriam Diamond systematically started raising rats in enriched versus deprived environments. And what she found was that the rats that were raised in enriched environments had thicker cortices, and the rats that were raised in more deprived environments had thinner cortices. That is, they got thicker or thinner based on the shrinking of synaptic connections in the deprived rats, while enrichment allowed those synaptic connections that had been made mostly at birth to survive. And this is the very last slide. It's a map of the cerebral cortex of a human brain showing 180 cortical areas. Up until recently, it was thought we had very fewer, but this is the recent work of David Van Essen. And it's a composite. It's studies from many brains, and he's put it together here. And what he's discovered is that there's a huge amount of variability in all these areas. In some people, this area might be bigger or smaller. This area might be bigger or smaller, different sizes and shapes. It's like a fingerprint on your brain. In fact, the most different thing about identical twins is their brains. And we still don't know what most of these areas do, but the fact that babies are born with all these areas, and they're very different, and that's partly due to their different genetic makeup. It's partly due to in utero influences. It's partly due to mother's experience. It's partly due to variability and random influences, which I discuss in the book, in various parts of the book. And now I'll quote from near the end, and this is what I'll end with. Everyone's brain differs from everyone else's from the moment of birth. And these differences only grow as the final shaping of our brains is done outside the womb and incorporates our individual experiences in the world. What makes us all human makes us all different. And with that, I'll thank you, and I'm happy now to take any questions if there are.
Thank you so much, William. That was wonderful, an amazing tour, and to me, actually remarkably poetic in terms of the story of life and death, and pioneers and travel and so on. So that was really wonderful. Thank you so much. So yes, as William says, there's plenty of opportunity to ask questions. If you'd like to ask a question, just pop it into the Q&A box. I can see one already, actually. So the question is, what is the most complex part of a baby's brain development?
The most complex part of a baby's brain development. I think they're all incredibly complex. Different parts of the brain develop at different times. So the spinal cord and the hindbrain are early developers compared to the cerebral cortex. So the cerebral cortex still has a way to go in terms of neuronal cell death and synaptic elimination after birth. But if you looked at any part of the brain at an electron microscopic level, it's just mind-blowingly complex.
Thank you very much. Here's another question that's really interesting to me, actually. When do we gain consciousness? At what age and to what extent? And for other animals as well. So
yes,
that's a big question there.
It's a big question, and it's one I tend to avoid because I don't think we all agree on what consciousness is. I would say that a baby in the womb has maybe brief awake periods that we've talked about, and in those brief awake periods, they may have some sense of life, or that they're a living thing or something like that. So consciousness, yes, it's something that is with us when we're awake, and maybe with us when we're dreaming in REM sleep in a weird way. Babies spend most of their time asleep, and maybe most of their time not very conscious, but they have brief moments of consciousness. And I won't want to talk about other animals because people still say, is a fly conscious or not? I would say it is, but not everyone would.
Thank you. Yes, it's clearly one of the biggest ongoing challenges that we have in this area. Right, the next question. Have any of the substances which guide axon growth been implicated in the neural migration disorders?
Oh, yes, that's quite interesting. I wouldn't be able to give you a number on that, but I am willing to say that neural migration, that is, when a neuron is born, it's usually born at a particular place in the brain, and it may migrate before it sends out an axon and a dendrite. And neurons use some of the same cues that guide axons. So imagine the early embryo has molecules all over it that you can read like signposts, and migrating neurons can read those signposts and migrate into an area. And when they send out an axon, they can read the same signposts to go to a different area. So that's still actively being worked on. But yes, it's very cool that the same molecules can be used for neuronal migration and axon guidance and dendrite guidance.
That's super, thank you. There's a question, really interesting talk. Why do so many tracts in the brain, such as in the optic chiasma and the decussation of the pyramids, cross? Is there an evolutionary purpose to the crossing?
Yes, people think so. I explained how the optic chiasm crossing helps to merge the right and left eyes, so that both eyes can, seeing the same thing, can go to the same place in the brain. But you need to coordinate activity between the left and the right sides when you're walking, when you're holding something, lots of coordinated activity. And it's good for the right side of your brain to know what the left side of your brain is doing. And in human brains, it's quite interesting that there's quite a lot of asymmetry between the right and the left side of the brain. And certain areas on the brain that we're seeing in this slide here, or that we saw in the last slide, they're bigger on the right side, smaller on the left side, and others are bigger on the left side and smaller on the right side. But they're talking to each other, and they talk to each other through these decussations from one side of the brain to the other.
Thank you. Here's a question about kind of environmental factors. So what do the results of the environment say about deprived inner city areas?
Don't know about that so much, but we do know that there are effects of deprivation that are transgenerational in the brain. Certainly, if you're food deprived, if a mother is food deprived during pregnancy, or if a mother is alcoholic, and recently, I think smoking has also been implicated in this, this will affect the way that the baby's brain grows. And it does that through mechanisms that we don't particularly know. One of them is very obvious. Is vitamin B9 that is involved in many cases of spina bifida, where the nervous system doesn't close up properly, and that is if you don't get enough of this particular vitamin, you have an increased chance of having a baby with spina bifida or something worse. So these factors, some poisons, some vitamins, and things like that, are all having an effect on the way that the brain is growing in the embryo. And as I say, they also affect the what we call the epigenome. That is not the genetics of the brain, but they make imprints on the DNA that can be read out over a lifetime. And it's debated now, sometimes over generations. And there's some story in the book about, how they taught mother rats or mice to fear an electric shock that was associated with a bell, and their babies who'd never really experienced that were also more sensitive to that.
Thank you. Thank you. I mean, one thing that came out of your talk very strongly was the fact that we need to, every neuron is crucial, and we must try and avoid losing them as much as possible. I mean, is there any advice you can give? I mean, you talked about a few things not to do. Is there anything that we can do to kind of preserve our neurons?
That's a good question. Don't get a neurodegenerative disease if you can avoid it. Don't get a crash bump on your head if you can avoid it. If you're playing rugby or something like that, then that's harmful. If you get bashes on the head. Exercise, they say, is good for mental health, but I don't know what its role is on brain neuron survival. But you're right, every neuron is in a particular place and doing a particular job. It's processing a certain bit of information that's going there and sending it on. So every neuron is doing something slightly different from every other neuron in terms of processing information. And you want to preserve them all, but I can't give you a mechanism to do that.
That's a shame, but understandable. Thank you. Here's another question. So can you tell us some interesting facts about brain degeneration, and has your research found things out that have aided work on brain aging?
I can say my work has not done that. It's not been about aging, it's been about development. However, what is important to think about, I think for people like that are in the field that I'm in, is that understanding how the brain develops might be useful in terms of medicines in the future for brain degenerative diseases. If we could get axons to grow again that have been lost through degeneration, if we could get new neurons born in the brain somehow, and have them be young and be able to navigate, then one might be able to repair some of the problems in brain injury and disease and degeneration.
Thank you. That's wonderful. Right, another question. So some people have apparently survived quite normally despite having very little brain matter, e.g. due to hydrocephalus. Please, can you comment on this?
Yeah, I suppose you can live without a whole visual cortex, but hydrocephalus changes the structure of the brain. It doesn't necessarily kill all the cells in the cortex. It squeezes it, deforms it, and the brain is very soft. They say it's like cream cheese. So as long as you don't kill the cells in the brain, hydrocephalus will distort the shape of the brain, but it won't necessarily kill the cells. But if you do lose critical areas in your brain, then you do lose functions. For example, if you lost your visual cortex, you would be pretty much blind.
Mm-hmm. Yeah.
And people with strokes that lose, the ability, the areas that are involved in language learning, as you know, they lose the ability to communicate.
Mm-hmm. Thank you. Okay, how much of the brain do we know we use? And are we using more now than a few hundred years ago?
I think we use it all. I think it's all there for a reason. And when people say, use 10% of your brain or something like that, I think that's weird, because it's all there. If you record from any part of a brain of an animal, you'll find times when that part of the brain is active. Now, certain parts of the brain are more involved in certain tasks, like the visual areas at the back of your brain are more involved in visual tasks, and the front of your brain may be more involved in planning what you're going to do tomorrow, and the language areas are involved in speaking and hearing and understanding, but you need them all.
I mean, there was part of that question about whether we use our brains more now than we did 100 years ago. I guess perhaps related to that is the idea that IQ might be growing over time as measured.
IQ?
IQ, yes.
Yeah, I don't know. That's another subject I kind of avoid, because intelligence means different things to different people. Some people are socially intelligent, some people are mathematically intelligent. One thing that's changed from 1,000 years ago, or maybe 10,000 years ago, is written books and things like that. So language has probably become more sophisticated, and it might be interesting to see if you ever could compare the language areas of prehistoric humanoids, hominids with modern ones.
Thank you. I think we've got time for just one more question. We can sneak it in. So, are the neural survival factors different to neurotransmitters? Is it a more complex process than just use it or lose it?
Yes, it is a much more complex process. And when I teach the subject, and I'm talking at the early stages of brain development, where we're talking about how neurons communicate with each other before they've made synapses, and how they're guided and so forth, and whether they survive, and they're talking to each other, transmitting survival. There's a lot of molecular give and take neurons, or they're communicating with their environment and with each other, adhering or not adhering, long before they're making synapses. And then when they make synapses, then they use these synaptic transmitters. And there's a bunch of those too, like glutamate and GABA, excitatory and inhibitory transmitters. But before then, and while the neuron is still alive, there's a lot of other factors that the neuron is paying attention to, such as survival factors.
That's great. Thank you very much. There are still more questions coming, but I think we must draw this to a close. So thank you again, William, for the wonderful talk and for answering all of these really great questions. Just a quick reminder that the recording of the event will be available on the festival's YouTube channel, which can be accessed from the Watch Again section of the festival. Go and have a look at the website and see if you can find some more great talks to enjoy. You will be contacted by email when the video is available to view. And of course, if you'd like to buy William's book, Zero to Birth, How the Human Brain is Built, it's going to be available from our partner booksellers, Fox Lane Books.